Canidae. The biological family Canidae is a lineage of carnivorans that includes domestic dogs, wolves, foxes, jackals, dingoes, and many other extant and extinct dog-like mammals. A member of this family is called a canid. The cat-like feliforms and dog-like caniforms emerged within the Carnivoramorpha 43 million years before present. The caniforms included the fox-like Leptocyon genus, whose various species existed from 34 million years before present, before branching 11.9 million YBP into Vulpini and Canini. Canids are found on all continents except Antarctica, having arrived independently or accompanied human beings over extended periods of time. Canids vary in size from the 2 meters long grey wolf to the 24 centimeters long fennec fox. The body forms of canids are similar, typically having long muzzles, upright ears, teeth adapted for cracking bones and slicing flesh, long legs, and bushy tails. They are mostly social animals, living together in family units or small groups, and behaving cooperatively. Typically, only the dominant pair in a group breeds, and a litter of young is reared annually in an underground den. Canids communicate by scent signals and by vocalizations. They are very intelligent. One canid, the domestic dog, long ago entered into a partnership with humans and today remains one of the most widely kept domestic animals. Taxonomy. In the history of the carnivores, the family Canidae is represented by the 2 extinct subfamilies designated as Hesperocyaninae and Borophaginae, and the extant subfamily Canini. This subfamily includes all living canids and their most recent fossil relatives. All living canids as a group form a dental monophyletic relationship with the extinct borophagines, with both groups having a bicuspid on the lower carnassial talonid, which gives this tooth an additional ability in mastication. This, together with the development of a distinct entoconid cusp and the broadening of the talonid of the first lower molar, and the corresponding enlargement of the talon of the upper first molar and reduction of its parastyle, distinguish these late Cenozoic canids, and are the essential differences that identify their clade. Phylogenetic relationships. Nyctea, Utes, Cuon and Canis within the Canidae. The results of allozyme and chromosome analyses have previously suggested several phylogenetic divisions. The wolf-like canids, including the dog, grey wolf, red wolf, eastern wolf, coyote, golden jackal, Ethiopian wolf, black-backed jackal, side-striped jackal, dhole, and African wild dog. The fox-like canids, which include the kit fox, red fox, cape fox, arctic fox, and fennec fox. The South American canids, including the bush dog, jory fox, crab-eating fox, a maned wolf. Various monotypic taxa, including the bat-eared fox, grey fox, and raccoon dog. DNA analysis shows that the first 3 form monophyletic clades. The wolf-like canids and the South American canids together form the tribe Canini. Molecular data imply a North American origin of living canidae some 10 million years ago, and an African origin of wolf-like canines, with the jackals being the most basal of this group. The South American clade is rooted by the maned wolf and bush dog, and the fox-like canids by the fennec fox and Blanford's fox. The grey fox and island fox are basal to the other clades. However, this topological difference is not strongly supported. The cladogram below is based on the phylogeny of Lindblad et al., modified to incorporate recent findings on Canis and Vulp species. Evolution. The Canidae today includes a diverse group of some 34 species ranging in size from the maned wolf with its long limbs to the short-legged bush dog. Modern canids inhabit forests, tundra, savannas and deserts throughout tropical and temperate parts of the world. The evolutionary relationships between the species have been studied in the past using morphological approaches, but more recently, molecular studies have enabled the investigation of phylogenetic relationships. In some species, genetic divergence has been suppressed by the high level of gene flow between different populations, and where the species have hybridized, large hybrid zones exist. Eocene epoch. Carnivorans evolved from Maya during the late Paleocene. Some 5 million years later, the carnivorans split into two main divisions, caniforms and feliforms. By 40 Maya, the first member of the dog family proper had arisen, called Prehistoric Canidae. Its fossilized remains have been found in what is now the southwestern part of Texas. The chief features which identify it as a canid include the loss of the upper third molar, and the structure of the middle ear which has an enlarged bulla. Prehistoric probably had slightly longer limbs than its predecessors, and also had parallel and closely touching toes which differ markedly from the splayed arrangements of the digits in bears. The canid family soon subdivided into 3 subfamilies, each of which diverged during the Eocene: Hesperocyaninae, Borophaginae, and Canini. Canini is the only surviving subfamily, and all present-day canids including wolves, foxes, coyotes, jackals, and domestic dogs belong to it. Members of each subfamily showed an increase in body mass with time, and some exhibited specialized hypercarnivorous diets that made them prone to extinction. Oligocene epoch. By the Oligocene, all 3 subfamilies of canids had appeared in the fossil records of North America. The earliest and most primitive branch of the Canidae was the Hesperocyaninae lineage, which included the coyote-sized Mesocene of the Oligocene. These early canids probably evolved for the fast pursuit of prey in a grassland habitat. They resembled modern civets in appearance. Hesperocyanines eventually became extinct in the Middle Miocene. One of the early members of the Hesperocyanines, the genus Hesperocyon, gave rise to Archaeocene and Leptocyon. These branches led to the Borophagine and canine radiations. Miocene epoch. Around 9 to 10 Maya during the late Miocene, Canis, Eurasian, and Vulpes genera expanded from southwestern North America, where the canine radiation began. The success of these canines was related to the development of lower carnassials that were capable of both mastication and shearing. Around 8 Maya, the Beringian land bridge allowed members of the genus Eucyanomines to enter Asia, and they continued on to colonize Europe. Pliocene epoch. During the Pliocene, around 4 to 5 Maya, Canis lepophagus appeared in North America. This was small, and sometimes coyote-like. Others were wolf-like in characteristics. Canis latrans is theorized to have descended from Canis lepophagus. The formation of the Isthmus of Panama, about 3 Maya, joined South America to North America, allowing canids to invade South America, where they diversified. However, the most recent common ancestor of the South American canids lived in North America some 4 Maya, and the likelihood is that there were more than one incursion across the new land bridge. One of the resulting lineages consisted of the gray fox and the now extinct dire wolf. The other lineage consisted of the so-called South American endemic species, the maned wolf, the short-eared dog, the bush dog, the crab-eating fox, and the South American foxes. The monophyly of this group has been established by molecular means. Pleistocene epoch. During the Pleistocene, the North American wolf line appeared, with Canis edwardi, clearly identifiable as a wolf, and Canis rufus appeared, possibly a direct descendant of Canis edwardi. Around 0.8 Maya, Canis ambrusteri emerged in North America, a large wolf. It was found all over North and Central America, and was eventually supplanted by its descendant, the dire wolf, which then spread into South America during the late Pleistocene. By 0.3 Maya, a number of subspecies of the gray wolf had developed, and had spread throughout Europe and northern Asia. The gray wolf colonized North America during the late Rancalabrian era across the Bering Land Bridge, there being at least 3 separate invasions, with each one consisting of one or more different Eurasian gray wolf clades. mtDNA studies have shown that there are at least 4 extant C. lupus lineages. The dire wolf shared its habitat with the gray wolf, but became extinct in a large-scale extinction event that occurred around 11,500 years ago. It may have been more of a scavenger than a hunter. Its molars appear to be adapted for crushing bones, and it may have died out as a result of the extinction of the large herbivorous animals on whose carcasses it relied. In 2015, a study of mitochondrial genome sequences and whole genome nuclear sequences of African and Eurasian canids indicated that extant wolf-like canids have colonized Africa from Eurasia at least 5 times throughout the Pliocene and Pleistocene, which is consistent with fossil evidence suggesting that much of African canid fauna diversity resulted from the immigration of Eurasian ancestors, likely coincident with Pleo-Pleistocene climatic oscillations between arid and humid conditions. When comparing the African and Eurasian golden jackals, the study concluded that the African specimens represented a distinct monophyletic lineage that should be recognized as a separate species, Canis anthus, according to a phylogeny derived from nuclear sequences. The Eurasian golden jackal diverged from the wolf-coyote lineage 1.9 million years ago, but the African golden wolf separated 1.3 million years ago. Mitochondrial genome sequences indicated the Ethiopian wolf diverged from the wolf-coyote lineage slightly prior to that. Golden jackal and dhole by A.N. Komarov wild canids are found on every continent except Antarctica, and inhabit a wide range of different habitats, including deserts, mountains, forests, and grasslands. They vary in size from the fennec fox, which may be as little as in length and weigh, to the gray wolf, which may be up to long, and can weigh up to. Only a few species are arboreal: the North American gray fox, the closely related Channel Island fox, and the raccoon dog habitually climb trees. All canids have a similar basic form, as exemplified by the gray wolf, although the relative length of muzzle, limbs, ears and tail vary considerably between species, with the exceptions of the bush dog, raccoon dog, and some domestic breeds of Canis lupus. Canids have relatively long legs and lithe bodies, adapted for chasing prey. The tails are bushy, and the length and quality of the pelage varies with the season. The muzzle portion of the skull is much more elongated than that of the cat family. The zygomatic arches are wide. There is a transverse lambdoidal ridge at the rear of the cranium, and in some species, a sagittal crest running from front to back. The bony orbits around the eye never form a complete ring, and the auditory bullae are smooth and rounded. All canids have digitigrade, meaning they walk on their toes. The tip of the nose is always naked, as are the cushioned pads on the soles of the feet. These latter consist of a single pad behind the tip of each toe, and a more or less 3-lobed central pad under the roots of the digits. Hairs grow between the pads, and in the Arctic fox, the sole of the foot is densely covered with hair at some times of year. With the exception of the 4-toed African hunting dog, there are 5 toes on the 4 feet. But the pollex is reduced and does not reach the ground. On the hind feet, there are 4 toes. But in some domestic dogs, a 5th, vestigial toe, known as a dewclaw, is sometimes present, but has no anatomical connection to the rest of the foot. The slightly curved nails are non-retractile and more or less blunt. The penis in male canids is supported by a bone called the baculum. It also contains a structure at the base called the bulbous glandis, which helps to create a copulatory tie during mating, locking the animals together for up to an hour. Young canids are born blind, with their eyes opening a few weeks after birth. All living canids have a ligament analogous to the nuchal ligament of ungulates used to maintain the posture of the head and neck with little active muscle exertion. This ligament allows them to conserve energy while running long distances following scent trails, with their nose to the ground. However, based on skeletal details of the neck, at least some Borophagini are believed to have lacked this ligament. Dentition. Skull. Most canids have 42 teeth, with a dental formula of. The bush dog has only 1 upper molar with 2 below, the dhole has 2 above and 2 below, and the bat-eared fox has 3 of 4 upper molars and 4 lower ones. The molar teeth are strong in most species, allowing the animals to crack open bone to reach the marrow. The deciduous, or baby teeth, formula in canids is molars being completely absent. Social behavior. Attacking a sambar, Bandipur National Park. Almost all canids are social animals, and live together in groups. In general, they are territorial or have a home range, and sleep in the open, using their dens only for breeding and sometimes in bad weather. In most foxes, and in many of the true dogs, a male and female pair work together to hunt and to raise their young. Grey wolves and some of the other larger canids live in larger groups called packs. African wild dogs have packs which may consist of 20 to 40 animals, and packs of fewer than about 7 individuals may be incapable of successful reproduction. Hunting in packs has the advantage that larger prey items can be tackled. Some species form packs, or live in small family groups depending on the circumstances, including the type of available food. In most species, some individuals live on their own. Within a canid pack, there is a system of dominance so that the strongest, most experienced animals lead the pack. In most cases, the dominant male and female are the only pack members to breed. Canids communicate with each other by scent signals, by visual clues and gestures, and by vocalizations such as growls, barks, and howls. In most cases, groups have a home territory from which they drive out other conspecifics. The territory is marked by leaving urine scent marks, which warn trespassing individuals. Social behavior is also mediated by secretions from glands on the upper surface of the tail near its root and from the anal glands. Reproduction. Canids as a group exhibit several reproductive traits that are uncommon among mammals as a whole. They are typically monogamous, provide paternal care to their offspring, have reproductive cycles with lengthy prostral and dioestral phases, and have a copulatory tie during mating. They also retain adult offspring in the social group, suppressing the ability of these to breed while making use of the alloparental care they can provide to help raise the next generation of offspring. During the prostral period, increased levels of estradiol make the female attractive to the male. There is a rise in progesterone during the estral phase and the female is now receptive. Following this, the level of estradiol fluctuates, and there is a lengthy diestrous phase during which the female is pregnant. Pseudopregnancy frequently occurs in canids that have ovulated, but failed to conceive. A period of anestrus follows pregnancy, a pseudopregnancy. there being only one estrus period during each breeding season. Small and medium-sized canids mostly have a gestation period of 50 to 60 days, while larger species average 60 to 65 days. The time of year in which the breeding season occurs is related to the length of day, as has been demonstrated in the case of several species that have been translocated across the equator to the other hemisphere, and experiences a 6-month shift of phase. Domestic dogs and certain small canids in captivity may come into estrus more frequently, perhaps because the photoperiod stimulus breaks down under conditions of artificial lighting. The size of a litter varies, with from 1 to 16 or more pups being born. The young are born small, blind and helpless, and require a long period of parental care. They are kept in a den, most often dug into the ground, for warmth and protection. When the young begin eating solid food, both parents, and often other pack members, bring food back for them from the hunt. This is most often vomited up from the adult's stomach. Where such pack involvement in the feeding of the litter occurs, the breeding success rate is higher than is the case where females split from the group and rear their pups in isolation. Young canids may take a year to mature and learn the skills they need to survive. In some species, such as the African wild dog, male offspring usually remain in the natal pack, while females disperse as a group, and join another small group of the opposite sex to form a new pack. Canids and humans. One canid, the domestic dog, entered into a partnership with humans a long time ago. This partnership is documented as far back as 26,000 years ago, when the footprints of a young boy aged about 8 to 10 were found in Chalvet Cave in southern France, walking alongside what was identified as a large dog or wolf. The earliest recorded fossil of a dog was found to be around 36,000 years ago in Goyet Cave in Belgium. Even earlier, wolves were found fossilized in the same locations as humans at sites that date back 300,000 years, showing how far back humans and wolves had interactions with one another. The fact that wolves are pack animals with cooperative social structures may have been the reason that the relationship developed. Humans benefited from the canids' loyalty, cooperation, teamwork, alertness and tracking abilities, while the wolf may have benefited from the use of weapons to tackle larger prey and the sharing of food. Humans and dogs may have evolved together. The bond between humans and dogs can be seen in the burial of dogs with their owners as early as 11,000 years ago in the Americas, and 8,500 years ago in Europe. Among canids, only the grey wolf has widely been known to prey on humans. Nonetheless, at least 2 records have coyotes killing humans, and 2 have golden jackals killing children. Human beings have trapped and hunted some canid species for their fur and, especially the grey wolf, coyote and the red fox, for sport. Canids, such as the dhole, are now endangered in the wild, because of persecution, habitat loss, a depletion of ungulate prey species and transmission of diseases from domestic dogs. Prehistoric Canidae. Except where otherwise stated, the following classification is based on a 1994 paper by Xiaoming Wang, curator of terrestrial mammals at the Natural History Museum of Los Angeles County, on the systematics of the subfamily Hesperocyaninae, a 1999 paper by Wang, together with the zoologists Richard H. Tedford and Beryl E. Taylor on the subfamily Borophagiinae, and a 2009 paper by Tedford, Wang, and Taylor on the North American fossil Canini. Subfamily Canini. Tribe Canini. Genus Canis. Canis adixis. Canis amagonoi. Canis apolloniensis. Canis Armbrusteri Canis, Arnensis Canis, Courtly Canis, Cedazoensis Canis, Donazani Canis, Scipio Canis, Ferox Canis, Gazi Canis, Lepophagus Canis, Mishoxi Canis, Mosbarkensis Canis, Neringi Canis, Variabilis genus Theriodictis, Theriodictis platensis, Theriodictis tarigensis, Theriodictis proplatensis genus Protocyon, Protocyon orsazi, Protocyon scagliarum, Protocyon troglodytes genus Cerdicyon, Cerdicyon avius, Cerdicyon onsonodensis genus Sputhos, Sputhos piscivorus genus Neurocyon, Neurocyon chonarchariensis genus Xenocyon, Xenocyon falconeri, Xenocyon lysaunoids tribe Vulpini genus Vulps, Vulps alepecoids, Vulps cf. Alepecoids, Vulps cf. Vulps, Vulps galaticus, Vulps referite basal canini genus Nightier Utes, Nightier Utes cf. Donazani Nightier Utes cf. Megamastoids, Nightier Utes, Donazani Nightier Utes, Megamostoids, Nightier Utes sinensis first. Canini genus Eucyon, Eucyon davisi, Eucyon minor, Eucyon joey, Eucyon monticenensis, Eucyon odesnus genus Leptocyon, Leptocyon vapor, Leptocyon vulpinus. Subfamily Borophaginii. Genus Archaeocyon, Archaeocyon falcon, Bachi Archaeocyon, Leptodus Archaeocyon. Pavidus genus Otarocyon, Otarocyon Cookie-Otarocyon McDonaldi genus Oxytocyon, Oxytocyon cuspidatus genus Rhizocyon, Rhizocyon oregonensis tribe Phlaeocinini genus Synarctoids, Synarctoids acridans, Synarctoids emery, Synarctoids gorni, Synarctoids harlowi, Synarctoids lama, Synarctoids luscensis, Synarctoids roii. Genus Phlaeocyon, Phlaeocyon acaros, Phlaeocyon inetans, Phlaeocyon latidans, Phlaeocyon leucosteus, Phlaeocyon marii, Phlaeocyon marslandensis, Phlaeocyon minor, Phlaeocyon multicuspis, Phlaeocyon taylori, Phlaeocyon yacali tribe Borophaginii genus Cormacyon, Cormacyon cope, Cormacyon haydeni genus Desmocyon, Desmocyon mathawi, Desmocyon thompsoni genus Metatomarctus, Metatomarctus canavus, Metatomarctus sp. A Metatomarctus sp. B genus Eulipocyon, Eulipocyon brachygnathus, Eulipocyon spicidans genus Sauridicyon, Sauridicyon marianae genus Microtomarctus, Microtomarctus conferta genus Protomarctus, Protomarctus optatus genus Tephracyon, Tephracyon rostris subtribe Synarctina genus Parasynarctus, Parasynarctus Kelloggi, Parasynarctus sinclairi genus Synarctus, Synarctus crucidans, Synarctus galushii, Synarctus marylandica, Synarctus saxatilis, Synarctus vorhii se, Synarctus 1g subtribe Aelurodontina genus Tomarctus, Tomarctus brevirostris, Tomarctus hippophaga genus Aelurodon, Aelurodon asthenostylus, Aelurodon ferox, Aelurodon magrui, Aelurodon montanensis, Aelurodon sturtoni, Aelurodon taxide subtribe Borophagina genus Paratomarctus, Paratomarctus euthos, Paratomarctus temerarius genus Carpacyon, Carpacyon compressus, Carpacyon limosis, Carpacyon robustus, Carpacyon webbi genus Protepicyon, Protepicyon raki genus Epicyon, Epicyon Aelurodontoids, Epicyon haydeni, Epicyon sevis genus Borophagus, Borophagus diversidans, Borophagus dudley, Borophagus hilly, Borophagus literalis, Borophagus auk, Borophagus parvis, Borophagus pugnata, Borophagus secundus. Thank you for watching. Please like and subscribe below. Please like and subscribe below.